Okay, good afternoon, everybody. Welcome to Friday's Biology Department seminar. It is my sincere pleasure to introduce today's guest, Wellington Muchero. He is a quantitative geneticist in the Biosciences Division at Oak Ridge National Laboratory, which is affiliated with the Department of Energy. And he also holds a joint faculty appointment in the Plant Science Department and the Breeson Center for Interdisciplinary Research at the University of Tennessee in Knoxville. Wellington received his Bachelor's of Science degree from Cal Poly Pomona and his PhD in plant pathology from University of California in Riverside. He also did a brief postdoc there before he joined the DOE lab of bioenergy and innovation in 2010, I believe. Wellington has made some major contributions to biofield research in quantitative genetics of trees and also in plant immunity. He's received millions of dollars in federal funding, including the prestigious DOE Early Career Award. Wellington has published extensively in many journals such as the Plant Cell and PNAS, New Phytologist, PLOS ONE, and many others. And he also has contributed to a few patents. So it is really my sincere pleasure to welcome you finally to UMass Boston, and I look forward to your presentation. Welcome.
Wonderful. Thank you so much, Aidan. And thanks everyone for joining us this afternoon. If for some reason my audio goes haywire, you may go ahead and let me know, Aidan, just in case. You
okay.
Never know with internet, yeah. But yeah, so it is my pleasure to join you today in Boston. I wish it would have been a visit in person, but I'm actually happy now that I'm joining you in 60-degree weather in Tennessee while you guys are freezing over there. So it's the most beautiful visit to Boston that I've had so far, I may say. I will share a little bit of our work today based on insights that kind of evolved after I joined the Oak Ridge National Lab in 2010, and where I feel like as a biologist that it's opening up new avenues for us to understand, especially genetics and how we approach genetic studies and what model systems we pick for understanding food production, fuel production, human heritable diseases, and how to intervene into that, managing disease, managing parasites, and things like that. So I will apologize in advance if the content of my presentation is bouncing you all over the place and it's not coherent and logical. It's just the nature of the work that we do. And I purposely made it that way to kind of give you a broad perspective of the discoveries that we are making and the connections that we are making across many different biological functions. So and acknowledge funding sources that have contributed towards this work. So these are DOE funded. The Bioenergy Science Center was funded by DOE from 2008 until 2016. And then subsequent to that, we applied for funding via the Center for Bioenergy Innovation, and that has been in place since 2017. And in addition to that, the Oak Ridge National Lab also provided us funding through a laboratory, a directed research program for us to dabble with human work that we do, because traditionally DOE does not fund work in humans. And outside of funding, acknowledge multiple collaborators that have contributed towards this work. Stephen DeFazio at West Virginia University, Jerry Taskin here at Oranel, Roba Bidea, she was a graduate student at Michigan Technological University, Jay Chen, who's my colleague at Oak Ridge, Chongo Pan, who has helped me with some work in bioinformatics, Renee Haps at the National Renewable Energy Laboratory in Colorado, who did some characterization for biomass that I'll talk about today. And Meng Shi was a postdoc in our group who did some of the work that I also talk about today. And last but not least, Emily B. Smith was an undergraduate intern who helped us actually develop some of the fundamental knowledge that we have since used in this project. And I had mentioned before when I was hanging out with students that we do host students at Oak Ridge National Lab. So if people are interested, you could be in the same position as Emily, and you can see how valuable those interactions are to us, because her work actually led to some fundamental science that we are still pursuing today. She's since now proceeded to graduate school at the University of Tennessee. I'll briefly introduce what the DOE mission is, what DOE is really interested in when they give us funding at Oak Ridge National Lab. And that is really to understand how changes at the DNA level. So when we look at genomes, we know genomes are made of DNA, and DNA is made of 4 basic nucleotides. And we know that a change at any position, at any nucleotide, could either have really fundamental impact on how that gene functions, or it has no impact at all. So our interest in the work that we do is trying to understand how mutations in genome affect gene function. So we're at the gene level. And then from then, how does that gene function affect the protein structure and what it does? Beyond the protein structure and what it does, what are the molecular interactions that are also impacted by those small molecules? So here we are still looking at really minute scale that we are looking at. And then those changes, we also know that they can manifest themselves in terms of cellular function of those genes, genes and proteins. Beyond individual cells, we know then that can impact how a whole organism can function. And then that organism is also functioning in the context of a community. And then from the community, you have a whole ecosystem. So what DOE asks us to do is look at what changes happen at the genome scale and what implications they have all the way to the ecosystem assembly of organisms. So this is a really complex question that involves many, many different disciplines of science to kind of get off. So in my position, the way we decided to go about answering some of these questions was using population scale genomics. And so what we did was we picked Populus trichocarpa, is typically known as black cottonwood. And it's a tree that's found in the Pacific Northwest of the US. In the East Coast, you have Populus deltoides, which is called the eastern cottonwood. It's a closely related species. And so what we thought to do was sample in the natural species range of Populus trichocarpa all the way from California. This is right at the bottom here. We are in like close to Lake Tahoe, and then all the way into British Columbia. And what you can see is the ecological range that has been represented by all individuals that were sampled in this case. And each blue triangle that you see, or each blue marker that you see, represents an individual tree that was sampled. So we visited that tree, took propagation material from that tree, and assembled a population of about 1,500 individual Populus trichocarpa genotypes. And then subsequently, we established 3 common gardens. We actually established a 4th one just this last year at the University of California, Davis. And these are all replicated. So each individual has 3 replicates per field site. And what that enables us to do is go in and assess how each individual performs. And we can look at individual replicates to see how consistent those observations are. And then we can also compare how consistent those observations are between really varying environments. Boardman, Oregon is right in the middle of the desert. So this is really hot and dry and sandy soils. And Corvallis is semi-dry, alluvial soils right next to a river system. And then Clascany, Oregon is really wet and muddy and clayey. And so in each environment, that same individual is experiencing very different environmental conditions. And we have the ability to then assign how that individual then behaves across environments. And apart from establishing the field size, the other thing that we did was then for each individual genotype, we worked with the Joint Genome Institute and sequenced completely every individual's genome. So for each and every individual genome, we know what mutations they carry relative to the reference. So for here, for example, I'm showing this is what a typical gene model would look like if you were to look at in our browser. So you can imagine that this is your string of DNA, and this is the predicted gene structure that's going to encode your protein, and you have non-coding regions, the coding regions in orange, and again, introns that are non-coding and things like that. And each then track that I'm showing here, 1, 2, 3, 4, 5 is the individual tree. So you can imagine 1,500 tracks like the ones that I'm illustrating for. And for each gene, we know what each individual, what mutations it carries across that entire gene. And in this example, I'm showing one where in this particular individual, you have a mutation that causes a frame shift. And so that frame shift affects how that protein is, how the mRNA is encoded and what protein it makes. And essentially what it does is it makes that protein non-functional. And then again, likewise in this individual, it has a mutation here, and you can tell the impact of the mutation by the color that is coded. Blue is really non-consequential. Orange is it has some impact, and red means it changes the impact of that protein in a very dramatic way. So we now have 1,500 individuals. Out of those 1,500 individuals, we have mutations at every single position of that genome. And just for reference, if you're familiar with genomics, the Populus genome is 480 million nucleotides. So you can imagine having this level of information on 480 million positions for each individual tree that you have in that study. The thing that we can then do is for those individual trees, we can go in and start taking measurements for every individual. And I'm only showing these kind of measurements because these are the primary ones that DOE is really interested in. Cellulose content for biofuels production, DBH and height and wood density is a measure of productivity. So we want to make as much biomass as we can per unit land that we planted those trees. And then lignin content, lignin is known as one of the most abundant polymers on the planet. You can use it for plastics. You can use it for many other things. It's also the one biggest challenge of making biofuels is you need to get rid of lignin before you can access cellulose in order to make a biofuel. So we need to know how to regulate lignin content in trees. And in each case, you can see that the distribution for each phenotype ranges quite a lot. So this is the natural variation for all those individuals, the 1,500 individuals. Some are high cellulose content, low lignin, low S2G ratio, high wood density, low density. And so you get a really big distribution of any particular trait that you measure in those individuals. Then the thing that we then are able to do, recall I mentioned we sequence the genome so we know what mutations are okay in each individual. And we have now collected phenotypic data, that's the traits that we are interested in mapping. And then we perform GWAS association mapping studies. And in very simple terms, what we do when we do GWAS association mapping studies is we are looking at each individual position in the genome. And here I'm giving an example in this particular individual, this particular genomic position. You have individuals that have this kind of mutation. And in this case, it happens to be a deletion. So no nucleotide at that position. And then you have individuals that are heterozygous. So it means they have one at that position. And you have a deletion, it has a deletion and a G nucleotide. And then in the alternate condition, you have individuals that carry both Gs that are homozygous for this position. And for illustrative purposes, this is the kind of variation that we see when we measure plant biomass. Exact same environmental condition, exact same care, same nutrients, same thing, and this individual grows by this much compared to this particular individual. So then what you can do is then classify your individuals by their genotype and say, if I look at individuals that have the deletion, what is their mean phenotypic score for that particular that we measured? And then if we look at the heterozygous condition, what is their average phenotypic performance versus the homozygous alternate allele, what is the average condition? And this is the kind of example that we typically see. Things get more complicated than I'm showing here for obvious reasons, just as a simple illustrative case. But in classical GWAS, this is kind of the results that we always see for most traits, where you see one mutation, individuals that carry one mutation tend to have a much higher phenotypic value compared to the rest of the population. And individuals that have the heterozygous condition are usually intermediate in terms of the phenotype that they express. And then lastly, individuals that the alternate allele usually have the lowest performance of that position. So in this case, we assume that this mutation somehow has to be related to that phenotype that we were measuring. If it wasn't related, then all these three classes would have the exact same phenotypic score. So we expect to have their average scores across the three classes exactly the same. The fact that individuals are different by their SNP mutation shows that that SNP is possibly affecting function of that gene where that mutation is found. So this is really the basis of gene mapping that we do in Populus. And as I mentioned, we have 1,500 individuals and 65 million SNPs that we do. So you can imagine doing this same kind of classification for each phenotype, for each individual across all 65 million positions that we have. So you can have 65 million plots like this that you can look at. And we are fortunate at Oak Ridge National Lab with the computational resources, world-class computational resources that we have. We can really do these calculations very quickly. It takes my postdoc probably minutes. Once I send him phenotypic data, within minutes, he comes back and it shows me the output that predicts what SNP mutation affect what particular trait. So we took this approach then to try to understand one key feature of plant genetics. So when we do bioenergy research, or at least any kind of plant research that you can do, one key component of that research is functional validation. And functional validation usually requires the regeneration of transgenic plants. That means you introduce foreign DNA into a plant cell, and you need that plant cell to replicate into 2 cells, into 4 cells, 8 cells, 16 cells, until you get enough cell mass of undifferentiated cells that you can then induce them to form a root shoot tip system. Then you are able to recover your transgenic plant. The initial part of that process is called callus regeneration. And here I'm showing just different qualities of callus, the way it might look in terms of size and shape and things like that. And I'll focus on this particular panel right now. And the white mass that you are seeing at the end of that little stem segment, that's what we call callus tissue. And we observed that across our population of 1,500 individuals, they varied greatly in terms of how much callus they formed. Some, as you can see here, they form huge amounts of it. And some in the exact same condition produce very little callus formation. So in a desirable situation, we want to have an individual that behaves like this, because this then facilitates transgenesis, as I'm illustrating in the bottom figure, where you start with callus going from panel A all the way. You can start seeing that now you're generating a little shoot system from your callus. And from your callus, then you form little root systems. And then now you have multiple plants that are growing from that same callus. You take those tissues, you put them in tissue culture, and from then you have your whole transgenic plant. This is how we essentially make plant transgenics. And so this process is essential to part. And so we wanted to understand, why do some individuals behave like this? And why do some individuals behave like that? And so we went across our whole population, measured this behavior, and as I illustrated in my last slide, did the correlation between callus formation versus mutations. And the first result that we found was kind of intriguing to me, and this is kind of where the rest of my story might make sense to you, was that we mapped that and found that mutations that promoted this kind of behavior were found in a gene that was annotated, and this is in the Populus reference genome. It's called a testis-specific protein, a SOC kinase. And that was kind of strange to me because we all know, clearly we know trees and we've studied trees, and testes are not a plant feature. Plants do not have testes. That's an animal, that's a mammalian feature. And so it was kind of intriguing to me, like, why is a tree gene given a mammalian gene? And specifically, this gene had been known to promote prostate cancer in humans. And not only that, there was evidence that actually this annotation for being called a testis-specific gene was actually the reason that it was first discovered, or the first group that discovered this gene and gave it a name was actually studying testicular cancer in humans. And they found that that gene was really, really critical in terms of regulating tumorigenesis in humans. And that was the first example. And then we looked again at the other candidates genes that were showing up, and it was the exact same story. The second one was the mitogen activated protein kinase that was involved in human tumorigenesis, exact same thing. The third one, XKLP2, progression in squamous cell lung cancer. The third one, an RPD3, an anti-tumor activity against human cancers. And what was intriguing about this one was actually this gene was highly active in individuals that did not form callus formation. And so I took a pause at this point in time and kind of looked back at it. And to me, now it makes absolute perfect sense. If you think about it, this is pretty much what tumors look like in humans. This phenotype in plants, what we call callus formation in plants, is uncontrolled cell division and uncontrolled DNA replication and cell division that gives you these masses. And so that's the equivalent of tumorigenesis in humans. So it's really not surprising that we are finding 4 different candidate genes that regulate that feature in Poplar that have already been implicated in cancer progression in humans. And likewise, the anti-tumor gene is being found in trees that do not form tumors. So that makes perfect sense. So it's repressing the ability of these cells to reproduce, for DNA to reproduce, and for cells to replicate in the manner that we see in this above example. So given this finding, I kind of sat down with my group and took a step back and say, what are the opportunities that we are seeing here? What is this telling us? And again, it came to me that we really shouldn't be surprised by that initial observation, because plants and humans are eukaryotes. We recognize that we shared a common ancestor way back in history, and we have since diverged and things look very different nowadays, but there are very fundamental processes that still have to remain similar. DNA replication and repair is still the same in plants and human cells. Cell division, cell adhesion, those things are fundamental to biology, and so they should be regulated in about the same way. And so we looked at this as an opportunity now to say, can we now look at the differences in plants and humans and say, of all those mechanisms that regulate these fundamental processes, what has not changed after 1.5 billion years? If we look at a protein that regulates DNA replication, a protein that regulates cell division, a protein that regulates cell adhesion, whatever has not changed are the key aspects of that protein that are required for its function. So that's where we came up with this idea to identify orthology of genes. So this means that sequences that are still similar in humans and plants. And then we know that only amino acids that haven't changed should be the ones that are essential. And so then we designed this study where we looked at genomes across eukaryotes, and this is coming from everywhere: Animalia, Heteroconta, amoebas, fungi, plants, protists. So we took all these genomes and I then designed a study where we look at what amino acids haven't changed, or what genes are still shared. In those genes, in the proteins they encode, what amino acids haven't changed, and can we start predicting function from that? But before we go into the molecular details, I'll share a few more examples of what we did again in the same populations to kind of illustrate that this concept is just not unique to the one example that I showed you. So here we were interested in understanding what forms a rough bark in trees. Some trees have rough bark and some trees have smooth bark, and they are the exact same species. You have seen all over the place. If you look outside your window right now, you will see this. And so we were interested for ecological reasons, because when a tree has a rough bark, we notice that it tends to promote ecosystem assembly. You have bugs that can hide in there, and then those bugs attract other, birds that feed on those bugs. So you tend to have a little bit more complex ecosystems when you have a tree that have a rough bark versus a tree that have a smooth bark. And in other instances, you have lichens that like adhering to smooth bark trees, and they don't do so well in rough bark. So just by the nature of the kind of bark you have on a tree, you can change an entire ecosystem. And you recall our mission from the DOE is to find mutations at the DNA level that affect ecosystem assembly. And this is a classic example where if you are able to identify the mutation that causes rough bark versus smooth bark, you can explain why lichens are found in other trees, but not in others. And you can explain why some trees promote, all kinds of insects and birds that then come and prey on those insects versus the smooth bark traits. And this is the work that Robert Bidier did at Michigan Technology University. And so what we found was, if you then look at a cross-section of these trees, is essentially what's happening in the trees that have rough bark is they have annual accumulation of the bark layer. So this is this current year's bark inside right now that's developing. This was the previous year's bark layer and the other previous year's bark layer. So these barks, those layers are stuck together. They don't upsize. And then what happens is the tree is expanding because bark tissue is dead, so it's not elastic. It doesn't expand. Then it cracks. And that's how you form the rough feature that you see when you look at the surface. It's actually multi-year layers of bark tissue that have accumulated and then subsequently split as the tree is expanding in its growth to keep up, just to give the volume. Individual trees that have the smooth bark phenomena only have this current year bark. So this is the equivalent of this inside layer here on this particular individual. So this tree actually does not, the bark layer does not adhere across years. Every single year it sheds the bark and then it starts anew. Next year, same thing happens. Last year's bark is upsized, and then you form a new bark layer. That's what we found in this particular mechanism. Then we did the statistical analysis again, quantifying individuals that have the rough bark feature versus the smooth bark. Look at the SNP mutation and say what SNPs are found, or what mutations are found predominantly in trees that form the rough bark feature. And that's what we did. And what Robert found was actually that there was a position on chromosome 1 that was highly correlated with the existence of the adhered bark, of the thick bark function. And then when we looked into her data and kind of trying to understand what are the genes that are actually found in that position, we found one that's called a desmosome junction protein. And these proteins have been implicated in humans already. And what they do is actually you have two neighboring cells that are illustrated here. And then you have your desmosome junction protein. And what you can see is they form like a link. So they link the two cell layers together. In the functional version of that, you have two cell layers that are now stuck. And that is the annual accumulation of bulk that I was showing you. When it's not functional, then you don't have those two cells adhered, and that layer can upsize, so it means it can slough off and shed off. And we thought that was pretty intriguing. And then looked into what other studies have been shown implicated in this. And we actually found that, interestingly, the exact same condition occurs in humans, where this is called keratoderma skin cancer. And it's a genetic defect where your skin layer, like the normal human being, our skin layer sloughs off every single day. So our cells do not adhere. So your skin cells do not pile up. In individuals that have a mutation in this protein, the skin actually does the exact opposite. So it doesn't upsize, so it keeps piling up. And so they end up having these masses of dead skin cells that look like wood tissue, and that's keratoderma skin cancer. So this condition is actually highly genetically analogous and is the exact same protein that does the same feature where you see rough bark in trees and keratoderma in humans, whereas this individual genotype in trees represents our normal human phenotype, where our skin sloughs off. So that was one example. And then the other example that we did was, when we do our studies, we need to sample wood as it develops, so we understand the genetic basis of wood development. And what we typically do is we cut out these windows in trees, and then we scrape off that mass, and what you see in that mass, that's developing xylem. And what ends up happening is we leave a scar on that tree. Then a year later, when we come back, we look at all trees and we see these three different behaviors. One, some of the trees have marginally covered up that scar, but that window is still wide open. So this is the equivalent of that you can see right here. Some have actually closed a pretty good section of that wound to only a small part of it is still visible. And on the extreme end, they have completely closed that wound. So they have completely healed what we had done to it at the beginning. So again, we measured that behavior, correlated it to mutations, and say what genes are regulating that behavior. And we found three particular candidate genes that were really exciting to us. And what was really fascinating to us was these genes had been highly studied in sea cucumbers. And if you know anything about sea cucumbers, is they have a really unique defense mechanism where if they feel threatened, they will puke out and so throw up their entire gut system to try and entangle whoever is threatening them. So here is an example where you can actually see all that you're seeing is their guts. So it throws up its entire gut system. And what's remarkable about sea cucumbers is then they are able to just sit back there and regenerate that whole gut system again. And off they go, and they go eating, and life returns to normal. So tissue regeneration is really unique, and sea cucumbers have adopted that as a defense mechanism. Throw up your gut system, entangle your predator, move away and regenerate it, and you're normal. And so we are seeing the exact same process here, where we are looking at tissue regeneration, and the same genes that sea cucumbers use to regenerate their guts are the main primary genes that we also found that regulate this behavior for where plants are actually able to really close off their wound. And not only that, this is the exact same mechanism that also implicated in humans. Humans have only two organs that regenerate, the liver and skin. And when we are hurt, we all have scraped our knees and elbows and things like that. Our skin is able to heal and close off any wounds that we have. This is the exact same behavior and the exact same genetic mechanism we are seeing in trees, sea cucumbers, and in humans. The other example that I would like to share with you, and this comes directly out of work that we do for bioenergy research. So the fundamental principle in bioenergy is you look at sugars in plants, and you can ferment sugars to make fuel. You can make ethanol out of sugars. humans have been doing this for, for all the wrong reasons. Moonshine, for example, is, you can make moonshine from corn because it has high starch content. And the basic principle is the same, is you ferment those sugars and then you have fuel. So one of our major goals is how do we increase glucose content in plants? Because the more glucose you have, the more fuel you can make, the more ethanol you can then make from fermentation. So we did this really study that I will not dwell on much and explain all these graphs to you. But again, for glucose, we saw that there was significant variation in individuals in how much glucose they actually contained in their biomass. So we harvest all the trees, ground up the biomass, measure how much glucose they have. And you can see some individuals have really low glucose content, some has really high glucose content. And then we correlate again back to our SNP mutations, see what happens. And what we found was actually the gene that primarily determines glucose content in humans is the exact same gene that regulates blood glucose content in humans. So the diabetic condition, people who have a malfunction of an alpha-glucosidase tend to have higher glucose levels, and we know that that's not, that is a detrimental condition. So we need to regulate the amount of glucose that's found in blood. And what we were finding was the exact same enzyme that regulates blood glucose level was the same enzyme that regulate glucose level in biomass. And then we looked at, so we know that condition, and here's an illustration of the differences or the similarities between the Arabidopsis version of that protein, the popular version of that protein, mouse and human. And you can see that they are pretty much essential. They are pretty much the exact same in 3 different protein domains, except for one domain that's only found in mouse and humans. Outside of that, everything else is identical between plants and humans. So the glucose level in blood, glucose level in wood regulated the exact same way. And although it's detrimental in humans, what we actually found was when we induce the diabetic condition in plants, those plants actually grow much bigger and much faster. So they grow much bigger, they grow much faster, and they have higher glucose, and that's exactly what DOE is interested in. And here I'm showing compared to the wild type, you can see that actually the knockout mutant for that alpha-glucosidase has a much higher biomass accumulation. So again, basic principle, regulating glucose is the same in plants as in humans. It's kind of like biology is that conserved. It uses the exact same mechanism, but in 2 very different systems. Blood and wood are completely different, but the actual protein that regulates those processes remains identical. So it's kind of easy to make these phenotypic correlations, but we have actually not shown any functional data to show that what we are claiming in some of these cases is true. So we picked an example where we wanted to do an actual functional validation study. And here we picked a condition that happens in humans. This is called the Divergent syndrome. And it happens when there's a deletion on chromosome 22. It's a partial deletion of chromosome 22 in humans. And you cannot have both copies of that gene deleted. So you need one functional copy. So this copy is still functional, and one of them is deleted. And what happens is then you get developmental abnormalities in individuals that carry that deletion. So it's called Divergent syndrome, and it comes with a whole host of associated defects, mental, developmental, all kinds. And here we just show a few examples. And what we found interesting was actually that humans have one copy of that gene, and plants also have one copy of that gene. And in Arabidopsis, we could actually delete both copies, whereas in humans, if you delete both copies, it's lethal, so the organism does not survive. We did a study in zebrafish where we knocked out both copies, and it was embryo lethal. Studies have been done in mice where you delete both copies and it's embryo lethal. So you can really not experiment on this gene using mammalian systems that are the typical medical research platforms. And surprisingly, in Arabidopsis, we knock out both copies. And compared to the wild type here, you can actually see that the deletion, 2 independent deletion mutants actually grow bigger. They're actually much happier than that. So we thought this was a very nice system for us, and it should allow us to understand the molecular function of this particular gene. And here, what I'm now showing you is how close humans and mice are. And this is why mouse are typically used for human studies, because they share such close relationship. So on a protein level, that human gene, the DGCR14 in human and mouse, are actually almost identical, almost 95% identity. So almost all amino acids are the same except for a few. Whereas when we compare human to the polar tree DGCR14, now you see that there are only few amino acids that remain conserved. Almost everything else has been mutated. So our hypothesis was, if these 2 genes were still to have the same function, and if we could demonstrate that they have the same function, then it's really easy for us to pinpoint what parts of those proteins are absolutely essential. In this alignment, you cannot do that between human and mouse because almost everything is the same. But in plants, you have eliminated a whole bunch of regions, and you're only now left with 1, 2, 3, 4 regions that look like you can actually mess around with. So if you are trying to engineer, then we can precisely lead you to positions of that proteins that are more likely to give you the preferred outcome. And here again, I show the difference between the wild type and the Arabidopsis that are lacking both copies of that gene. And you can see even in developmental that both individuals that are deleted actually grow much faster, they flower much faster. So we can grow more plants, we can harvest seeds and then go through the next generation much faster than we can do with the wild type. So this was a very nice system for us to now start asking the question, do we have the same function between plant and human? So the next thing that we did then was we took the Arabidopsis gene, and so we have the knockout mutants that lack both copies. And in this case, we are looking at now, how does the Arabidopsis gene behave, or the Arabidopsis protein and the human protein behave in the exact same cell? So we expressed both variants, and what we saw was expressed in Arabidopsis, the plant version behaves like this. So it's actually localizing in the nuclear compartment, and it shows that punctate behavior where you see that spotting. And we express the human variant, it shows the exact same behavior. And here we are showing that they both localize in the same compartment. So we take a human protein, express it in a plant cell, it still knows how to get to the nucleus. It still knows how to integrate into the spliceosome complex, as we are showing here, exactly like its Arabidopsis counterpart. And here is just a zoom in of that image where you can see the Arabidopsis and the human protein behaving exactly the same way in exactly the same cellular compartment. Then we look at, we had implicated this gene that it actually binds to mRNA, and it's implicated in mRNA processing. And we could evaluate and see what are the shared targets. When we introduced the human, some divergences happened, but what we were able to show was they were actually able to target a very conserved amino acid DNA sequence in both plants and humans. And so this is the Arabidopsis variant and the sequence that it targets. And this is the human variant and the sequence that it targets. And those sequences are almost identical. So after billions of years, the Arabidopsis and the human DGCR14 protein still know how to behave and how to target the exact same DNA sequence. They still behave exactly the same way in how they localize and how they form a complex when expressed in the same cell system. So then the key question is, what happens when we mess around with some of those amino acids that have been conserved for billions of years, right? Naturally, like I had said in my original thoughts, was we expect that should have huge consequences because those amino acids have remained the same for billions of years for a reason. So to address that question, we mutated one of the conserved domains targeting these 3 amino acids right here. And we converted them randomly to a KKK in the plant and in the human variant of the same protein. And then we compared them to the unmutated forms of the plant and of the human. And as you can see, both the plant now lose their punctate, that spottiness that they have when expressed in plants, and the human in the exact same manner also loses its ability to behave in the same way compared to these are the unmutated variants. So that exact same mutation has the same implication whether mutated in the plant version of DGCR14 or in the human version of DGCR14, showing that these 3 amino acids are actually essential for incorporation of the DGCR14 protein into the spliceosome complex, as we are showing here. This is the normal behavior that we would expect. This punctate, yellowish and reddish in the nucleus right there. And when we mutate only those 3 amino acids, both the human and the plant completely lose that behavior. And now they're just diffuse in that whole system and they lose their function. So this is actually demonstrating that we are right in stating that these highly conserved amino acids are essential for function of those proteins. But the proof is, again, is always in the pudding. So how do we demonstrate this on a phenotypic level? And our postdoc was really smart in this case. Meng Shi did this work. And he found that the DGCR mutant was highly susceptible to salt stress. So what happens is when you add salt to growth media, they tend to die out. And when they die out, you see they look like these bleached whitish plants right here compared to surviving ones. So, and then compared to the wild type, the DGCR14 mutant was much more susceptible to salt. So this is kind of a nice test phenotype that we can say. And the question that we were asking was, if we take the Arabidopsis mutant that lacked that gene and put back the Arabidopsis via transgenesis, put back the DGCR14 gene, can we rescue that behavior? So can we recover salt tolerance? And this panel here is showing what happens when we take the DGCR14 mutant and put the DGCR14 gene back to it. Now we see that it's behaving a little more normal in terms of its salt tolerance. It's a little bit better than wild type actually, because these were overexpression lines. But what was even more exciting for us was when we took the human gene and put it in Arabidopsis and did the exact same experiment. Now you see it's even much better in terms of its salt tolerance. So these complementation assays are actually telling us that the human gene, even though it has been apart from plants for a billion years, when we put it back in Arabidopsis, it does the exact same thing that the Arabidopsis counterpart does. So this is kind of the proof in the pudding that we have been waiting to just kind of demonstrate that what we were showing was not just statistical artifacts based on a genome-wide association mapping, but there's actually a functional basis that proteins from plants and humans still do carry the exact same function. And that's why we were seeing those similar phenotypes that I explained to you earlier. And that story kind of took an interesting turn when we started looking at immune responses. This is getting close to the end of my presentation. And again, one of the key questions that we are interested in bioenergy is how do we manage bioenergy plantations without loss to disease? And disease is one of the biggest causes of loss in productivity in bioenergy scenarios. And we were looking at a particularly damaging pathogen called Septoria stem canker. And it's like anytime you plant these trees, within a year, they develop these giant cankers from these fungus, and the trees just collapse, as you can see right there. So you cannot grow these trees, the black cottonwoods, in eastern United States for that reason, because that disease is so highly prevalent. And what was interesting was we found in one particular case that individuals in our population that were highly susceptible to this particular pathogen always expressed one kind of gene, and it's called the G-type receptor like kinase. And so this gene actually promotes infection. Instead of defending the plant, it allows the pathogen to enter and cause disease. So it's a susceptibility locus. Then we are also interested in how to manage plant productivity by beneficial microbes. And we know plants, especially perennial plants, associate with fungi that are called mycorrhizal fungi. And these mycorrhizal fungi actually infect the plant root, and they enmesh themselves in a way that they are able to exchange nutrients. So they get the fungus get carbon from plants, and in exchange, it feeds nutrients to the plants. And so this is essential for phosphorus, especially phosphorus mining, nitrogen exchange, water exchange in terms of managing drought and things like that. So these beneficial interactions are actually highly essential for us to be able to, for plants to survive in the way that in the natural environment. You can imagine a plant lives for 300, 400 years on the exact same spot. It cannot move. So how does it explore the surrounding environment? The best way is to find other organisms to interact with. So then they do the exploration and bring nutrients to you. And then in turn, you give them carbon. This is the mycorrhizal interaction that happens between plants and humans. And we were able to find a gene actually that regulates that interaction. So if you think about it, this is the wild type plant that does not carry that gene. And the green spots that you see are the fungi. The purple are the plant cells. And when we introduce that gene, again, you see now that that fungus is actually able to make it way inside the plant, inside the plant cells. So again, akin disease that I mentioned, this gene again allows the fungus to invade and infect the plant. So this was in one kind of poplar that we did the test. We did a test in another type of poplar, exact same thing. This is without the gene. Before you engineer the gene, you still see mostly plant cells, very few fungi are found within the plant. When we introduce the gene, it's like all kinds of that fungus, and you can see it forms like gloves on the root system of the transgenic plants compared to the wild type. And so again, we are demonstrating that this gene is essential for colonizing. And what's essentially happening between these and the mycorrhizal story that I told you about is immunosuppression. So you are preventing host from triggering defense responses where in the natural condition, organisms fight off invaders. In this case, these genes are actually allowing the invaders to come in. And so we were interested in what is that behavior. Long story short, we were actually able to pinpoint one protein domain that functions in immunosuppression. And then outside of plants, what we were able to demonstrate was actually that one domain that I talked about is also implicated in fertility. So this is in Arabidopsis, where Arabidopsis are self-compatible, and so they are able to self-fertilize, and you get really healthy seed production. So that means that individual does not react to its own genetic material, so you have compatibility. When we knock out that gene that carries that protein domain that I'm talking about, that we saw in the pathogen and in the mycorrhizal example, now you have loss of fertility. So it's like the same plant has become allergic to its own pollen, so it can no longer be self-fertile. And not only that, we found that that same domain, protein domain, actually is found in human sperms. It's actually found on the egg side of things, where it functions as a sperm receptor. And if you think about it, for an egg to be fertilized, it has to mute its own immune system against the sperm. Otherwise, it will have an allergic reaction and you get infertility. It's the exact same protein domain that mediates that function. Not only that, we found that it was also the same domain that explains how Daphnia magna is a water flea and is very renowned for harboring a very complex ecosystem in its gut. So it has many organisms living inside its own body. And we found that it actually uses that same domain to mute its immune reaction so it doesn't reject those organisms. They actually come in and thrive. That's one example. The same protein domain is found in nematodes that infect humans. So again, humans naturally fight off pathogens, the human body system. But when you have that protein domain in nematodes infecting humans, then those nematodes are actually able to infect humans, and you get elephantiasis, as you see in this particular example. That's outside of that. And then insect vectors of disease. We know mosquitoes vector malaria, Zika virus, and Lyme's disease, and all other kinds of parasites. So those parasites have to reside inside the mosquito body before they are transmitted. And the exact same protein domain that we found in plants explains how mosquitoes are actually able to carry those parasites. And here, last on this part, I'm showing psyllids. So psyllids infect, they carry the citrus greening virus. That's causing all kinds of devastation, especially in California right now. Exact same mechanism. They use that protein domain to mute the immune system of the psyllids. That virus lives inside the insect. When the insect feeds on a tree, it transmits that disease. And akin to the symbiosis that I talked about, nodulation of legumes. We know legumes carry beneficial bacteria that do nitrogen fixation. Exact same process. And lastly, in humans, again, cancers proliferate because of lack of immune regulation. In that particular domain, one of my postdocs has actually finished studies human cell lines, and I'm not going to show that research today, but where we showed that that domain actually is essential for cell proliferation. So when that domain is functional, you get tumorigenesis. Your cells just go out of whack. When you mutate that domain, you can actually prevent cell proliferation and stop any kind of cancerous growth. So in summary, I know I've shared a lot of information, and I'll do this really quickly. We came up with the conclusion that we actually can use plants as model systems to understand eukaryotic biology. And for many, many reasons, one of the major ones being in plants, we need much smaller populations to do statistical association studies. These kinds of studies in humans typically take hundreds of thousands to millions of individuals to get significant associations. In plants, we are doing this with 300, 400 individuals, and our 1,500 panel is actually much more than we need. And we get much higher resolution to the point where we can identify exact mutations that affect what traits occur. And then plants tend to maintain higher levels of genetic mutations. If you can imagine yourself in winter next year, what you'll be seeing is pollen flying around. There's pollen everywhere. And so pollen lands on a compatible a stigma, and you get fertilization. And that's how mutations happen. So plants are non-selective in their sexual behavior. If you think about humans, we are very, very picky about who we fraternize with and how many kids we want to have. And that limits how much mutations can spread within human populations. That's why you need millions of individuals to do association studies. It's just that our behavior changes the way mutations can carry across populations. And then for some reason, we seem to show evidence that plants can tolerate some of these mutations, like I showed in that deletion, that in mammalian systems, human, zebrafish, mice, it's embryo lethal. But in plants, we can delete both copies and actually get bigger and more and faster growing plants. And not least of which is plants are really easy to experimentally manipulate. You can imagine doing the experiment that I illustrated with the DGCR14 using animal models. That's super complicated. And for us, again, fewer ethical issues. We didn't have to get any sort of special permits to manipulate plants. We went around cutting out wounds in plants, and you could never do that in animal systems. So you can do things to plants that you could never do in any animal system. And what our evidence is showing us is at least in those cases where the orthologs are still highly conserved, we still end up with as much information as we would have if we had used animal models. So we are highly hopeful that this gain traction, and we can start using plants more and more to understand human and animal biology and vice versa. And with that, I would like to acknowledge many individuals that contributed to this study. I couldn't have done this by my own self, but they are so numerous to mention. So I thank you for your attention, and I'll be happy to take any questions now.
Great, thank you very much, Wellington. Maybe we can have one quick question, but considering the time, I'm wondering whether we could also switch over to the happy hour. But maybe one quick question, and then we could switch over to the other Zoom location.
There's one in the Q&A already.
Okay, great. This is from Rick. Maybe you said this, but I missed it. For rough versus smooth bark, the question for poplar, I would imagine that's what he means, would seem to be, is this polymorphism neutral, or do one or the other of these alleles provide some fitness advantage to the tree? Do you know?
So that's absolutely correct. So it looks like it's a neutral mutation, depending on context. The rough bark mutation tends to be very beneficial when you are growing in really dry areas. So it looks like it's related to drought tolerance. But the smooth bark tends to be associated with trees that grow in riparian systems, so along river systems. So it looks like trees need to lose water more quickly in those environments. So depending on the environment, either one of the allele is actually beneficial. So it seems like it was an adaptation event to dry versus wet environments. And so you need one variant over the other in each environment.
Great. Maybe we could continue the discussion at happy hour, and maybe I'll give you a couple of minute bio break or something, and then we'll join you. I put the link to the happy hour on the chat here as well for anybody who wants to join us, and maybe we can see you in a couple of minutes there.
Yep, sounds good.
Okay, thank you very much. See you soon, everybody.
Yep, thanks everyone.